Therefore, the title of my talk is Disposable Sensors for Next Generation On-Site Testing. And this is, again, 1 year later talk. I'm still a junior research group leader in Freiburg, so that didn't change in the last year, but we have new and exciting results, which I would like to share with you. So starting with maybe at the basics like we did last year, why disposable sensors? Because they are affordable, single-use and easy to handle sensing devices for short-term, let's say up to 2 weeks, or single-shot measurements, and they can transduce biological, physical, or chemical changes in their environment to an analytical signal, which we can measure. And they are so popular right now because these types of sensors are allowing us to mine critical information by anyone, anywhere, and at any time without worrying about the contamination as well as the recalibration. And they are, of course, already in our lives. So as a pregnancy test, they are known since, I would say, almost the 80s as a single-use devices where you can see yes or no answer. But if you want to be more sensitive, then you may need a readout device which is non-disposable. So they could be combined with a non-disposable device to increase the sensitivity and their usage. So and in this case, this is an evaluation test, which can be used as a digital reader together with the same lateral flow assay. There are, of course, also some electrochemical ones, and the most prominent ones are the electrochemical glucose monitoring systems. And here I choose an sugar watch, but this is usually also the same if you have a sugar measurement device. Currently, of course, the electrochemical systems already proven their capability also for wearable sensing, where they could be used also on our body as a wearable device, where the readout can be done by an external device, so which you can see here, and then all the results can be read out with smartphones. Maybe you can ask yourself why it's like that. So I'm just going very deeply, since if as a diagnostic company, if you want to make it use of a smartphone for your sensor, then you need to test all different types of smartphones out there. But with such a device, if you have your own device, you only need to validate this device to use. So what we are doing here in Freiburg, what's the current research, what we are doing? Of course, multiplexing is very important for us. Not only one analyte or one type of sample we would like to process, we would like to measure simultaneous analytes or different types of samples at the same time. And here we can measure either antibiotics, inflammation markers, or nucleic acids. Another area where we are still working on, of course, is variable immunoassay systems. Here we are aiming at a variable immunoassay device for hormone detection on freely moving birds, and the whole system will be located within this reflective jacket or coat, so that the chicken does, and there is a continuous blood administration, I would say every 2 hours, and then the chip, there will be an on-assay chip incubation as well as a signal readout electrochemically. The next thing is an OptoAssay, which I won't go much in detail because today will show you the first preliminary results of this study, where we are using the light regulation and reversible interaction of optogenetic switches to create a dynamic bioassay. And finally, which I won't speak today that much, is the paper-based breath sensors. Here we use paper, the functionality of a paper as a porous material to carry the electrodes as a substrate, but also to immobilize biomolecules, as well as for containing salts, and getting the humidity of our exhaled breath allows us to do electrochemical measurements on the simple, let's say, dry paper. And of course, finally, it's a sampling method, which is non-invasive. So here we are working currently on a glucose detection, but the results are not premature yet. So maybe next year, if I'm here again, so we will hear it again. But let's start again with the last time's highlights with the CRISPR-powered diagnostics. CRISPR can be used as for the nucleic acid detection in two types of, in two different modes. And the first one is a binding-based CRISPR diagnostics. And I think today also my colleague, Kiana Aran, will also speak about this CRISPR chip, where they use this binding-based CRISPR diagnostic, but this is a Cas enzyme which is deactivated for the cleavage. And so they just look at, for example, some genetic mutations in the whole genome. And if the Cas finds it, it binds and results in electrical change that you can detect with, for example, as a graphene field effect transistor in that case. In our case, we are working on the cleavage-based CRISPR diagnostics, where a target is recognized by the CRISPR-Cas system, and it starts to cleave all surrounding RNAs except the target in its environment. And this is also how we use it to create an RNA sensing method, which we published in 2019, the first electrochemical CRISPR biosensor. For this, we use an Cas13 enzyme with a CRISPR RNA, which you can, the blue part of this CRISPR RNA is complementary to your target, which means if you want to target another analyte, another nucleic acid, then you only need to change this part. We also use a reporter molecule on one side with an fluorescein, and on the other side biotin. And the only thing what you need to do is mix them together with your sample and leave it for a specific time at a specific temperature, which is depending on your enzyme that you are using. If there is no target, the CRISPR-Cas system does not get activated, and you can create, for example, like we did in our single analyte channel, you can create a bioassay where you can combine, use, for example, glucose oxidase as an enzyme to convert glucose and generate hydrogen peroxide, which you can detect later on electrochemically. What happens if there is a target? CRISPR-Cas gets activated. I'm always imagining by myself like the Pac-Man goes through this epi and cuts all reporter RNAs. And in that case, the biotinated site still can bind to surface, but the signal generating part is washed away. So we are assuming an competitive assay behavior. So if there is an analyte, then the signal lowers. And why CRISPR also within the last year gets so popular and also used, and there are lots of publications right now. The system is very simple, and you have also a time-dependent cleavage, so that if you wait longer, you get a better result, higher sensitivity. And also, if you want to change your analyte, the only thing that you need to change is on your system is this CRISPR RNA. The rest of it stays, which is also on the sensor, stays the same. So with this, we show that highly sensitive, specific detection. We show that we can program this for different RNAs. We show that we are fast and cheaper than qPCR devices. We validate our system with clinical samples. We show that on-chip cleavage is possible, and we came up to the multiplex analysis. And this is also, of course, shown last year in our recent publication in the Biosensors Bioelectronics, where we measured 2 different microRNAs simultaneously on a multiplex chip. But based on this result, we move forward now for a CRISPR-powered COVID-19 testing. And here our goal is multiplex amplification-free detection, less than 30 minutes. And we are targeting a first-line screening and envelope gene E, and confirmatory assay are the RNA-dependent RNA polymerase gene for the SARS-CoV-2, and as a negative control, SARS-CoV-1, and as a positive control, we use already validated microRNA as a target to show that the system is working. And here are the first results. So within the last year, thanks of my students, we were able to improve our design. So we have a new design of our CRISPR biosensor X, which has a vertical channel where we can get rid of some dead volume of the channel, and we can go 4, 6, and 8 analytes And we already measured the RDRP. We made a little RDRP calibration on a multiplex channel where you can see we are more sensitive than 1 picomole within 30 minutes. So if you already hear my talk last year, so our detection limit was about 10 picomole for 3 hours cleavage. Now we have a total sample to result time 30 minutes coming up less than 1 picomole as a limit of detection. So this is still preliminary results, and we are hoping to bring out a preprint very soon. We already also tested different genes of the SARS-CoV-2 in the same as we proposed. So 19B microRNA as a positive control that the system works, E gene and RDRP to show that the patient has SARS-CoV-2, and we have SARS-CoV-1 as a negative control. And we did the same, for example, for a SARS-CoV-1 patient. And here we have the otherwise situation. So we have here the 19B shows that the test works, and the patient has SARS-CoV-1 in that case, and SARS-CoV-2 genes are some kind of, in that sense, negative controls. So this is the first results finally that we could bring on, but the main highlight is to cut the 4 hours sample to result to 30 minutes, and by lowering also the LOD by 10. So this is and really huge success, I would say. But if you are interested also for doing CRISPR diagnostics, so what are expecting us in the future? So we are moving now to the point of care. We are aiming amplification-free detection, because unless you combine this with an isothermal amplification, it's not anymore that attractive, in my opinion, because you need again primers, probes, and different stuff, so that we need to be amplification-free regarding the nucleic acid side. So this is one of my messages. Although we already presented different genes on a single chip from the single sample, we could also, I think still multiplexing is an issue, so we need to be multi-omic. What about not only measure if the patient has COVID or not, for example, looking for the immunity, or checking if there is some drug monitoring, if there is some change in the drug clearance of the patient. Or, of course, another thing which I will think which will be important in the future is this CRISPR-based systems works very good for the nucleic acids. Why we cannot use them also for detecting proteins and maybe combining aptamers? So this is also the second part, which I think this is important. And of course, the next stage of the CRISPR diagnostics will be the wearables. And here we wrote this year also an article how the wearable devices could be used already for COVID-19 testing. And we saw also some couple of months ago by the group of Jim Collins from MIT, the face mask, which is combined with CRISPR to detect COVID-19. I think on Thursday, Jim will also explain about it a little bit more. So this is the CRISPR stuff, what we achieved within a year, but let's move another area. And this is where we are also active, is the therapeutic drug monitoring. So therapeutic drug monitoring, mainly we get a drug and prescribe with a dose that is tested on different small group of people, and this is used always the same. And here there are some limitations to be able to make personalized therapeutic drug monitoring. And these are the detection techniques like chromatography or immunoassays, which is related with high instrumentation cost, complex sample preparation, and long turnaround time. So we would like to see the clearance, so we can't wait a day for that. So and here we proposed also last year how sensors could be integrated into these devices. And our goal, in our projects, is to enable a personalized antibiotherapy. And we started with the first point, of course, management of the therapy, how we can dose, for example, beta-lactams. The next step will be assessment of the therapy success by inflammation markers. So is the therapy works, and do we need still to get the antibiotics further? And the next step then will be prognosis, like the organ functionality, like maybe renal functions, how they are affected due to the therapy. So for this issue, we just actually 1 month ago, I guess, published our latest study in Advanced Materials this year, where we propose a system for multiplex on-site therapeutic drug monitoring of antibiotics. Here we made an animal study with different dosed antibiotics, 50, 100, or 150% of the antibiotic is given to the animal. We studied different sample types using, again, a penicillin binding protein system as a competitive assay with the ampicillin biotin, and we have avidin GOX glucose oxidase as our enzyme, again, to create the signal. And here are the results. So we said we would like to look at the blood-based samples. Why is it possible to offer a solution like the blood glucose measurement to enable an antibiotic therapy? The next thing was we studied different multiple non-invasive samples and look, can we quantify drugs in these samples and how these are correlating with the blood results? And of course, finally, we go for a multi-analyte measurement where we try to detect 2 different analytes simultaneously, but also multiple samples. So could we measure plasma together with, for example, saliva or urine? So here are the results. We studied a whole blood measurement, which is just diluted whole blood, which we measure. And what we can see in overdose, underdose, and also normal dose case is we can clearly see and clearance behavior independent of the dosage regimen. So we could measure whole blood also as a matrix for our antibiotic measurements. The next thing was, of course, what about the non-invasive samples? And one of the first ones which I would like to look at was also exhaled breath condensate, because we have this mask application where this as a sensing methodology, and therefore we would like to see if we can detect in exhaled breath condensate antibiotics. This was already shown, but they just would able to detect something. There was now no quantitative detection. So, and what we here see in our case is we see that there is a correlation between the blood plasma values with the antibiotics, which means we can see a clearance in the exhaled breath condensate, which is great, so that we can use it. And this is also following the plasma values, because this is important. Of course, we studied also other samples like saliva and also urine in that case. And here we, as expected, actually, we see the history of the drug transportation. So it was a cumulative information rather than instantaneous feedback. So, and this is of course important. If you go for wearables, you need to have an instantaneous feedback. So the next step was, again, to see if we can measure multiple analytes at the same time. For this, we used another version of our multiplex chips, which we called Biosensor-X. And here we studied different piperacillin tazobactam together with the meropenem on the same chip before the administration of the antibiotics and 5 minutes later, where we expect the highest dose. And the results can clearly demonstrate, as expected, that the signal goes down after 5 minutes of the administration of the drugs. What we also would like to see, to enable maybe to study different samples simultaneously, we studied different analytes on the same chip: urine, saliva, exhaled breath, and plasma. And here we also look up before the administration and 1 hour later for these results. And as expected, we see in all cases again a decrease of the signal. But the next step was, of course, it's good to measure with your sensor, but you need to benchmark your system with the golden standard. And this is what we were also aiming and which we did with our single analyte chip, versus HPLC measurements for piperacillin tazobactam. And here you can see the results of these clearance signals. And what is important, so we see an similar exponential decay of both methods. And so the results are validated to be in a good accordance with the gold standard. We also did an internal validation of our single analyte chips together with the multi-analyte chip with the Biosensor-X, which is also shown here, and they are also fitting together. So on the multiplex side, we still need to some advancements to have the perfect chip at the end, but we are working on right now. But I think this is the part of the on-site therapeutic drug monitoring results. And on the last part of my talk, I would like to go and introduce a new assay design, which we called an OptoAssay. This is the first light-controlled dynamic bioassay using optogenetic switches. Here we are using the binding of phytochrome B to its interacting factor by only changing the wavelengths. And so our goal is to enable a wash and pump-free bioassay where we could enable a bidirectional movement of biomolecules only by changing the light. And for this, we studied different designs, and we came up as a best solution where we have a sender in the outer part and an inner part, a receiver from the top view. And if you are using it, there is a sample solution on top of these nitrocellulose. And so since the sample it comes to the top of it, we will have an contact of these two areas, and we start our system to regulate by changing the light. So maybe a little bit more explanation. Here we have the initial state where we have the sender, where we have a nitroavidine biotin, which keeps the Phoebe on the surface, and we have this PIF6 combined with an his-tag GFP, in order to enable us a signal change. On the receiver, we have an anti-his antibody. So we would like to show the proof of principle. So and by just detecting the his-tag in the middle. And so in that case, we have 660 nanometer wavelength. So the system is not, they are bound together. What happens if we go to the release? So we gave the sample and we release our competitor. And these two then goes and binds and competes for the areas in the receiver in this part by where we change the wavelength. What is the next step is to rebind the non-bound stuff back to the phytochrome B. So this is the idea, to have it only by the light to manage your system without using any external pumps or anything, or washing steps. So how it look like? So again, I would like to say this is really the proof of principle study of that. So first of all, we go for the release and looking up the competition. So here, maybe just to a little bit explanation. So this is our sender and this is the receiver. And if we have a low analyte, then we expect that the receiver receives the release stuff, and so we expecting a change of the signal in optical signal intensity. And in case we have a high analyte concentration, this part stays not that more lighter than the other area. So let's look at that. So this part needs a little bit more explanation. So here with red, you see the samples which are constantly stayed on 660 nanometers wavelength. So here we always made a duplicate. Here you see the sender and the receiver. and this is the after 40 minutes illumination. So as expected here, nothing really released on that. And we have, so here you can see also before and after of this signal. So there is no change. In the case of 740 nanometers, where we made the release of our competitor before the illumination, there is, of course, again, we see clearly the similar behavior like 660. But what happens if there is no analyte in the solution that we provided, then the receiver receives, of course, binds to the GFP with his tag, and so we see a signal increase, and on the sender side we see a signal decrease. What happens if we add an analyte? Again, at the beginning, as expected, it's similar to the other ones, but after we change the wavelength to the 740, then we see the sender again, the intensity lowers, but the receiver, since we gave also very high concentration of his tag biomolecules, so it's lighter than these signals. So this is the main idea of that. So we also studied the supernatant, so the solution which we just suck out to see if there is a difference. And as expected in the supernatant, we don't see much of the GFP, but with the analyte we see the highest concentration of the competitor, which is released to our signal. So we could show that the release works and the competition is working again also. The next step was, of course, not only to release and compete, so because this would be very easy and such a systems already exist, so we need to make another thing. So we need to look up if we can rebind the non-bound GFP histag molecules back to the sender. And this is again the same story. Here we have before, after, and here again only by 660, which again delivers us similar results here. Then we have the part with 740, and here we see that before the illumination there is no bonding, but after the illumination we see a binding here, and on the last one we see the release and the rebinding. And here we see that it's at the beginning all are some kind of similar, and here what we see, it's getting higher. So some parts of the molecules can bound it back to the sender area again. And the similar result we see also on the supernatant. Again, here we have the lowest signals where we do nothing at all, so we do not change the light. Here we have the release, so there is lots of in the supernatant inside, and on the bound back solution we see lower than the only released stuff. So here I need to, of course, before I finish this part, maybe I need to say this is the first proof of principle study. So we need to now think about it, how we can increase the release efficiency, how we can increase the competition efficiency, and as well as how we can improve the rebinding, because at the end we would like to get rid of a rebinding effect. So it should be only controlled by a smartphone. So, and here the Photobox that we designed for this purpose. So we have a mobile phone and a 3D printed box where we have the red and far red LEDs together with a smartphone camera where we can take the photo. But of course, for the GFP measurements, we did not use this setup, but we can improve this. We are going to now improve this setup also for the GFP measurements. So this is again a proof of principle study, not that advanced stuff that we are usually doing, but it's, I think, important because, of course, this is the first time that I'm working on an optical system, and I think this is also a very fancy for now, but maybe for future, after coming up, increasing the efficiencies of rebinding, it could be a really great tool to be used with optogenetic switches. And it could be also extended by using other optogenetic switches, combining to create more complex assays. With this, I'm end of my talk, but I have some slides. Of course, here, this is my group, so Disposable Microsystems group here in Freiburg. And so I need to wait so that you can see the nice city with the church background. So I would like to thank all of my, of course, group members and also collaborators, especially Jaren for the antibiotic measurements, also Regina for the antibiotic measurements. They are also present today. And Regina has also a poster, I hope, that where she will discuss how she did the multiplex chip design, and also for Midori to the CRISPR measurements, and Nadine for the great effort on the OptoAssays. So thanks for this. Again, after 1 year, I also improved my ending a little bit. So science is about to change knowledge and information. So therefore, let's get connected. You can find me in the social media under these channels if you want to be connected. And finally, we have still a wearable biosensors and bioelectronics special issue opened in the journal Biosensors Bioelectronics, where I'm also associate editor. So happy to receive your submissions if you are working on this field. With this, I'm end of my talk.
Ken, thank you very much. The mind almost boggles it, how much you managed to get into such a short presentation and how far you've advanced this technology since you were talking to us last time. So we are open for questions. We have a little bit of time left. So if any of our attendees want to ask a question, they can either do it through the chat line or through question and answers. Somebody has already. Did you use the microfluidic system to synthesize the multiplex microchips?
If I get the question some kind of right, so we are talking about most probably these chips. So they are containing a microfluidics. So this is, but the synthesis of we did not do the CRISPR or any cleavage here on the channels. So it's done again in the epi. So maybe on that, maybe I need to go back a little bit. Maybe like this would be easier. So if we go back here, so it's still the cleavage is done on an epi, but now we had actually shortened our cleavage time from 3 hours to 5 minutes. So and getting the same results and even better. So that's. And I hope to explain more once the preprint is there, but I'm hoping this time that it will be soon.
Okay, you had one comment. Somebody said, thank you for your amazing presentation, which I guess we all agree with. Let me see. Okay, another question is, what material is the chip made of?
In this case, for our microfluidic approach, we are using polyamide. So it's a polymer-based chip, which we actually, this is just a printed circuit board material, which we use also for producing the sensors. And also the channels are creating by dry film photoresist.
Okay. Any other comments? We have a little time left.
Michael, I've got a question maybe.
Yes, sure.
Okay. Yeah, no, I mean, it's always good seeing all the advantages. So tell us the downside. What are the challenges?
Of which one?
Well, just of using CRISPR-Cas
actually, the down advantage is really, we need to, from the sensing side, you need to be really sensitive. So again, I am telling, at least my goal stays to get to not to use the isothermal amplification methods. And in that case, we still are in a, now I would say, in a picomolar range. Maybe we are touching right now also 100 femtomole or a little bit higher. But still the sensitivity is an issue. So, and we need, and if I need to be also honest, so if I don't want to have an isothermal amplification, I need to find a better solutions from the sensing side to increase the signals. And this is also one of my next tasks.
So when you know, when you're thinking of isothermal or PCR, you often think in terms of copy number rather than picomolar.
Mm-hmm.
So what sort of sensitivities do you see in copy number?
Here we are, if I'm not wrong, 100,000 copies per microliter.
Yeah. Okay.
So this is just to be on the same on explanation. But regarding the viruses, what you could do, you can target 10 genes together. So which means you have 10 times higher concentration in that case. And this is also shown by Jennifer Doudna's group, where they came, I think, down to 1,000 copies or something.
Okay.
Yeah, and you know, obviously multiplexing would not happen in a single pot, would it? You'd have to split into multiple pots for this.
Right now, yes, but this could be possible with this technology here. So because I think this could be possible before I'm saying something wrong. No, it should be separated, right?
Yeah.
So in that case, we need to, either we need to say we are measuring different, not CRISPR combination with something else, but or we need to mix them together. But this is also possible. So we could also target E and RDRP, which will be also on the preprint as two targets for the same analyte to get a lower LOD.
Yeah, no, very good.
But I think it will be around 10,000 copies per microliter is something which is realistic, I would say, with an easy to use system to be able to detect without, right now, isothermal amplification.
Yeah. So it's got a way to go.
Yeah, sure.
Yeah.
I had one more question for you, Can. Did you use the carbon for the working electrode?
For this, no. Actually, we are using platinum electrodes, which we just evaporate on the polyimide substrate. So we use only carbon for the paper-based approach, where we are aiming at the targets from the Excel bread.
And a couple more questions have come in. Is it possible to have an industrial high throughput production of the microfluidic chip?
From this one, yes, definitely. That would be, I think, possible. Right now, we are facing actually another problem. So the dry film photoresist that we are using was produced by the company DuPont, and they started to, due to the material problems, they started to cut the line off. So and so as a scientist now we are working on different methods to create these channels, and we have already some ideas and we already started. So if this works, then it will be much more easier than the current version to produce in a higher numbers of these wafers.
Okay. And of course now we've got in Germany we've got microfluidic manufacturers in government, haven't we?
Have we?
You must know who I'm referring to.
Okay, no, actually not. But okay. So
one last question then, Can. Could it be possible to integrate the multiplex system on a digital microfluidic platform?
Definitely. I think multiplexing is something which can be done with various designs. So either you can do it with parallel channels, or like we do here with one single channel, which it has some compartments, different compartments. So actually, there are lots of techniques. I'm sure there are some methods to create microfluidics also for that.
Okay, well, Ken,
No, so Can, you must know that Holger Becker was elected to
Okay, I know him, but I didn't know that he's elected. Okay, oh, good.
Yeah.
Then I know who is it. Okay, maybe we will have some microfluidic boom in Germany now. So,
Ken, thank you very much.
thank you, Michael,
Brilliant
for the invitation.
contribution.
Thank you,
I
John, for
hope
the
we will see you again next year, and you can
I
take
hope.
the next stage
I
of the
really,
journey.
I really like to be here and looking forward to the next talks.